Okay, so I will start. So good morning, everyone. I'm Hayo from OTR GPQR. So today I would like to talk about a technique. It's called asymmetrical field flow fractionation, or we just call it AF4. Because it's too long the name. So this AF4 technique is a very advanced separation and characterization technique. And so it's a relatively new technique compared to HPLC, but there's an increasing use for the analysis of nanoparticle and the polymer material in the past decade. And also the pharmaceutical companies start to realize the value of this technique and start to use this technique. Some of them use the for their preclinical study to improve their formulation, and some company use in their for their generic product, to generate the data to demonstrate the comparable physical chemical properties. So today, the purpose of this presentation is give you like a general idea of this, what this technique can be used, and the capability of this technique. So after the a little bit background of and the principle of this technique, I will use a series of examples to show you the what can you do with this technique. And so before we go into any details, I would like to discuss a question. Is as I mentioned, this AF4 is a separation and the characterization technique. So some of you may ask, why do we need separation? Why can we just directly measure the samples? Of course, surely you can. For example, here you have so for example, here you have a nanoparticle standard of 10 and 30 nanometers. From the TEM image, you can tell, they are fairly uniform in size and shape. And we can use DLS to measure the particle size. And you will get a distribution like this. And we can see two separate peak when they measure individually. So, but if we mix the 10 nanometer and 30 nanometer in a 1-to-1 ratio and measure them again using DRS, instead of seeing two separate peaks, we'll get a distribution like this. And if we overlap the result of this mixture with the individually measured particle, we will see that the distribution of the mixed particle is very close to the 30 nanometer. So, as already took the training for DLS yesterday, you may know that this is because of the scattering of bigger particle is much stronger than the smaller one, and the bigger one kind of overshadow the smaller one. And the DLS, in general, does not have very high resolution. And so, this will, make us to think that, do we have other technique? And, of course, we can use TEM. TEM is a very high resolution technique that they image and measure individual particle. But as you can see here, depends on the uniformity of the samples. For a highly uniform sample like this particle standard, you may need to only count, a few hundred particles to get a meaningful distribution. But, in reality, we rarely have a sample like these particle standards. In reality, the particle are always like, the image shown on the right is some consumer product containing nanomaterial. The size span is very wide and is highly polydispersed. And for this type of sample, if we use TEM, we may have to count, thousands of even tens of thousands of particles in order to get a meaningful distribution. And that is very, labor intensive and time consuming. So that's probably one of the reasons why we want to, have a separation and a characterization technique. So the purpose of separation is that if we have a mixture of particles of different size, we use the separation technique to separate them into a few fraction. and each fraction contain relatively uniform particles. And then we use a different technique detector to characterize this individual fraction. So in the general idea is to to improve the resolution. We know the particle have many different physical chemical properties, and so it depends on which property do you use, and for the separation, the mechanism might be different. For example, you may separate a particle based on their mass or density, separate them based on their thermal diffusion, separate them based on their surface charge difference, or simply separate them based on their size. And of course, depends on the separation mechanism we may have many different techniques that can be used for this purpose. So today I'm going to focus on one category. Is called field flow fractionation. It's a type of liquid-based separation technique that the sample are separated in a field and in a liquid stream, and depends on what is used to generate this separation field. It can be further divided into a few subcategories. And so our main topic today will be the asymmetrical field flow fractionation. So first is, what is the AF4? It's actually a chromatography type of separation, but compared to traditional column chromatography, which they have a stationary phase, the AF4 has an open tubular design. It does not have stationary phase for the sample to interact with. So all the samples separation, in the liquid phase, and the liquid flow will use as a pseudo stationary phase. And the sample in AF4 are separated based on their hydrodynamic size, smaller particle, bigger particles. And the separation range for in AF4 is very broad. It's like 1 nanometer to up to 50 micron, but the most useful range is 1 to 1,000 nanometer in the normal mode. For much bigger particle, there's another mode called the steric or the lift hyperlayer mode that separate a particle in the 1 micron to 50 micron range. So let me first talk about some of the component or the instrument of the AF4. So in general, the AF4 contain a liquid chromatography system, a separation channel, as you can see from the bottom left, and a flow control module that control the liquid flow. And so you can see that the LC responsible to inject the sample into the separation channel, and the sample is separate there, and then they will pass through a series of online detector where you can get information from different aspects of the samples. And the right bottom is a picture of our system at DPQR. And you can see it actually is used the Angeline HPLC system, and the main difference compared to traditional LC is that it's a separation channel and a flow control module. So let's take a look at the core part of AF4, where the separation happens is the separation channel. Here is show you a disassembled separation channel, separated into different layer. From bottom to the top, we have a bottom block. On top of the block, we have a stainless steel frit. It acts as a physical support. So this frit is a highly porous structure, and it allows everything to pass freely, including sample and mobile phase. On top of the frit is ultrafiltration membrane, and it acts as a physical barrier. So, anything that is smaller than the pore size of the membrane, including mobile phase and some small molecules, can just pass through the membrane and go to this cross flow outlet. So if the sample is larger than the pore size of the membrane, and so it will be retained by the membrane. So in between the membrane and the top block is we have this called the spacer, and as you can see, there's a trapezoidal shaped opening in the center of the spacer, And this is actually where the liquid flow goes and where the separation happens. And so actually all the separation happens within this very thin layer. It's around only 300 micron. And here I show you some pictures of actual channel we use for the experiment. And it depends on how much sample you want to load, what is the purpose of analysis, what is the resolution you want to achieve. We can choose a different type of channel. So next, I want to talk about how the sample are separated inside the AF4. So as I said, the AF4 can be considered as a chromatography type of separation. Actually, a lot of the equation to calculate the retention, the resolution, the capacity are almost the same as the column chromatography. But in the traditional column chromatography, the sample separations start as soon as they enter the column. But in AF4, there's a unique step. It's called the focus, injection, and a relaxation step. This is the where it's quite different from column chromatography. And let's take a look at how this step works. So in this step, you have flow coming inside the channel from both inlet and outlet. And for the you have a stronger flow going from the outlet and the relatively smaller flow coming from the inlet and goes to the cross flow. And because the stronger outlet flow, the overall flow is pushed to the left, closer to the inlet. And in that case, it forms some like a focus flow closer to the inlet, and there's a focus zone. When a stable focus zone is established, we can open the injection port, where the sample that's stored in the injection loop is flushed into the channel. As soon as the focus and injection steps start, another phenomenon also takes place. It's called a relaxation. So I want to emphasize that this relaxation is not a separate step from the focus and injection. It happened at the same time when the sample is injected. So the sample is injected into the channel and stay in this focus zone. And so they are pushed down to the bottom, close to the bottom of the channel under this strong cross flow. And so the sample concentration that closer to the bottom of the channel is higher. And we know that sample tend to diffuse against the concentration gradient. So they will diffuse from a high concentration zone to a low concentration zone. So in that case, we know the Stokes-Einstein equation describes the diffusion of the sample. For smaller particle, and they tend to have a stronger diffusion coefficient. So smaller particle, they diffuse further away from the membrane and stay close to the center of the channel. Bigger particle, they have smaller diffusion coefficient, so they stay closer to the membrane. And of course, anything that is smaller than the pore side of the membrane will go to the cross flow and to the waste. And here I show you a picture of that when we inject a color sample into the channel. So as you can see that during the focus and then the injection step, the sample are confined into this very narrow band, and the elution will start after that. So this is the big difference compared to column chromatography. If we have a very dilute sample and we inject a very large volume in the LC, we will get a very big peak dispersion. But in AF4, we actually can concentrate this dilute sample in a narrow band, and then let the elution start after the sample is concentrated. So the next step is the elution. So the liquid flow in this step is different from the previous injection and relaxation. And to be noticed that is the actual channel thickness is only 350 micron, and the sample in general stay closer to the 10 micrometers away from the membrane. Here, all these are not to scale, kind of exaggerated to show you what actually happened during the separation. So in the elution step, that we have a stronger inlet flow that part of the inlet flow goes to the outlet, and part of the flow go to the cross flow and to the waste. And so in that case, the flow inside the channel will form something like that, something like this. You have a flow that continually exits from the bottom of the channel that, it will push down the sample to the bottom, just like that, what the sample experienced during the focus step. Also, there's another, is part of the flow is also flushing everything out to the outlet. So you have called the channel flow, and the channel flow has a parabolic profile where the center of the flow is faster, and the channel flow closer to the top and bottom of the channel is slower. So as I mentioned, because of the diffusion of particle, the smaller particle tend to diffuse further away from the membrane and stay closer to the center, so where they will experience a faster channel flow. In that case, they will be eluted at an early time. Bigger particle, because they stay closer to the membrane and they will experience a slow channel flow, so they will stay longer inside the channel and they elute at a later time. This is how we separate the particle based on their size. And when the particle finally eluted from the outlet and pass through a series of on-light detector, we will have obtain something like this, where you have peaks for each size fraction, and we have a particle size information, concentration information. And so for AF4, it has a very high resolution. Particle with a smaller size difference can be almost baseline separated. So the AF4 is actually a quite versatile technique. It's not just used to analyze nanoparticle. It can be used to analyze peptide and protein, study the aggregation, used for polymer material, and for particle used to study a variety of different particle samples like liposome, emulsion, metallic, metal oxide nanoparticles. And by using AF4, we can get a very comprehensive information on the sample. We can know what is the particle size distribution, their molecular mass distribution, what is the concentration of each fractions, and what is the shape of particle. Is there a spherical shape? Are they elongated? And what is the composition of particles? So next, I'm going to use a series of examples to show you the capability of this AF4 and what information can you obtain from this technique. And so the first example is ophthalmic emulsion. This is a cyclosporine ophthalmic emulsion. So as you can see, it's a kind of a complicated system that it has many components in there, including a big globule, oil globule, small oil globule, micelle, surfactant, dissolved component, and many more. And we know that the manufacturing process may affect the globule size distribution, and the formulation with different globule size distribution may have different efficacy. So to understand whether there's a difference in the efficacy, we need to accurately determine the globule size distribution. And for that measurement, in most of the cases, people tend to use DLS. I mean, we already know that DLS is not a high resolution technique. We use it to measure this ophthalmic emulsion. We'll get a Z average of around 110 nanometers with a PDI of 0.3. And so this DLS apparently is strong bias to the presence of big oil globule. And we can also use cryo-TM. Seems like a cryo-TM can capture the presence of small globule, but it seems to miss some of the presence of big oil globule. So if we use AF4 to characterize this formulation, as you can see from the right fractogram that by separate the globule into small fraction, both the big and the small oil globule can be captured by this technique. So we can use this technique actually to study the generic product and compare them with the RLD. As you can see here, we analyze several different products, ophthalmic products. Apparently there are difference in the amount of small with big oil globules, and this online size detector will give us size information, and the concentration detector will tell us how much sample in each fraction. And by combining the information from these two detectors, we will get a particle size distribution, like the figure in the right bottom. Apparently that not all the generic product are the same as the RLD. Actually, the generic B, apparently the distribution is quite different from the Restasis, from the generic, from the RLD. So the AF4 can give us very rich information for the particle size distribution. So beside the size distribution, what more important is the distribution of the API. Because of this ophthalmic emulsion, that the drug in the micelle phase or the API in the aqueous phase, their availability might be different than the API in the oil globule. So to assess the equivalence of a product, we wish to know that how much in each phase, and actually we can use AF4 to assist the determination of this distribution. So how we do that, is follow some process like this. So we know that the oil globule is covered with a layer of surfactant, and we know that there are excess amount of surfactant in the formulation. So beside the surfactant on the oil globule and dissolved in the solvent, the rest of the surfactants, because their concentration is way above CMC, therefore micelle. And in the formulation, we know the amount of oil in the formulation, and if we know the size of the oil globule, we can calculate the surface area that available for the surfactant to attach. So beside the surfactant on the surface and in the solvent, the rest of them will be just micelle. So, but there's a problem there, is because of the depends on the size of the oil globule, their total surface area will be different. And so that, for smaller globule, and the total surface area will be bigger. And so there will be less amount of surfactant in the formulation to form micelle. For bigger oil globule, it's the opposite. And so in order to accurately know the distribution, that we need to know very precisely the globule size distribution, and this cannot be done without a high resolution technique. And the AF4 is just the right tool for such purpose. So beside the emulsion, another complex product that containing nanomaterial is that we frequently encounter is the liposome formulation. And actually, AF4 is a perfect tool to determine the particle size distribution of a liposome. As you can see here, that from our internal study, we compared the particle size distribution of Doxil with a few generic doxorubicin liposome formulation and some in-house formulation. Apparently, their particle size distribution are different. And actually, that we see some, lot to lot or batch to batch variation for some generic product. As you can see, this red and blue color distribution are not the same, and they are from the same generic company. So AF4 has a very high resolution. It tells us a lot about their actual particle size distribution. And a unique thing that for liposome is that they depend on whether you loaded drug inside the core. For you may have empty or filled liposome. So if we use, a technique like DONS to measure, and it's very difficult to know whether the liposome is filled or empty. And we actually can, know all this information by using AF4 in a single run. And here is I show you the AF4 analysis of liposome formulation. One of the formulation is empty, the other one is loaded with drug. And from the the AF4, the top right, top left is we can tell that they have different hydrodynamic size distribution. And even for this purpose, it shows that the AF4 is much better suited for determining their particle size distribution. As you can see, the DLS did not reveal much difference. So in addition to hydrodynamic size, the AF4 can tell you another type of size. It's called the RMS size. As you can see from the top right, the RMS size for these two formulations are different, but we still don't know which one is empty and which one is loaded. And but by using AF4, actually, we can determine that. So here is a little bit more information regarding the RMS size. The equation in the top center is called the mean square radius. And the RMS is the root mean square radius. It's simply the square root of a mean square radius. So mean square radius actually describes the mean distance of every single atom in your particle to the center of mass. So in that case, if we have a hollow sphere, all the masses of all the atoms or all the mass are distributed on a thin shell of this particle. So the RMS size is actually quite close to the actual radius of the particle, equals to A. And we know that in a lot of cases, the hydrodynamic size is also very close to the actual radius of the particle. So for solid sphere, their hydrodynamic size will be the same, but their RMS size, which using RG as a symbol, is smaller because in addition to the mass on the outer layer, you have mass all the way to the center that causes a smaller RG size. So in that case, if for a hollow sphere, the RG to RH ratio is 1. For a solid sphere, RG to RH value is close to 0.7. And we actually can utilize this ratio to understand the conformation of particles. Now we combine the hydrodynamic size with the RMS size and get a plot on the top right. And we can see that the pink color formulation, the RMS to RH ratio is equal to 1. That means that the pink color formulation is the empty liposome. The green color one is a ratio of 0.7. That means that it is loaded with drug. So beside the knowing that whether a liposome is loaded with drug or not, that we can do more with the AF4. For the doxal formulation, a lot of time that because of the presence of this spindle-shaped doxorubicin crystal inside the liposome, not all the liposome particles are perfectly spherical shape. Some of them are elongated. So in that case, both their hydrodynamic size and RMS size may change. And we know that the doxal rely on passively targeting to deliver the anti-cancer drug to the tumor site. So if we have two formulations that is Q1, Q2 the same, but different in their shape, are they going to have the same targeting efficiency? To answer that question, we really need to know the actual size and the shape of the formulation. So by using AF4, we can separate the particle in the formulation based on their hydrodynamic size. At the same time, we can determine their RMS size. And by plot the RMS size with the hydrodynamic size, we'll get a shape factor. We'll get a ratio, which is a shape factor. Here is from some internal study results. And we study many different liposome formulation. Some of them are loaded with drug, and some of them are empty. Some of them contain a general spherical shape. Some of them are elongated. As you can see that, AF4 is very sensitive in picking up the difference in their conformation and tell us what is the shape of the liposome. So far we talk a lot about the the particle size and the structure of the formulation. But in a lot of cases that if we use the nanoparticle or like liposome or solid lipid particle as a drug carrier, we know not all of them are actually loaded inside the carrier. Some of them may be dissolved in the solvent. Some of them may be loosely bound to the carrier. So in that case, we want to know, okay, how much is loosely bound and how much is firmly loaded inside the carrier. And by using AF4, we can, study that matters. So in this case that, we inject the sample into the channel, and this instrument setup is quite similar as a focusing step. And, under the continual applying of cross flow, the API that dissolve in the solvent and the API that are loosely bound to the surface of the carrier will be, washed out from the carrier and go pass through the membrane and go to the cross flow port. And next we switch the liquid flow and flush everything out to the channel and pass to the detector. And by monitoring both of the liquid outlet port and the crossflow port, we can know how much is passed through the membrane and how much is stay with the carrier. Here is a very simple example of showing the encapsulation efficiency of a polymeric particle carrier. As you can see that there's a very good agreement between the amount of pass through the membrane and the amount of that to stay with the drug carrier. So far I talk a lot about the using AF4 for nanoparticle analysis. So in actually that AF4 has also been used for study polymer material, including biopolymer and synthetic polymer. So in fact that in a regulatory submission, I've seen, company use AF4 in studying, protein and peptide drug to understand their aggregation status. So we know that, high molecular aggregate is a big issue in the protein and peptide drug. They may have some immunogenicity problem. So knowing the amount of the high molecular aggregate is very important. And the AF4 is very good at, measuring large aggregates. And, as you can see that we use AF4 to study the, stability of protein and peptide drug under different stress condition. The top right, the aggregate with molar mass as big as tens of million Dalton can be easily characterized by AF4. But if we use SCC in this setting, it probably limited because that the exclusion limit of SCC column that we may not get very good separation for the very big aggregate. So the bottom two figures I pulled directly from ANDA submission. So the product is liraglutide, is a peptide drug used to treat diabetes. And the sponsor used both AF4 and SCC to study their aggregation studies. As you can see that while the molar mass distribution obtained by AF4 seems to be reasonable, there's we don't really see difference between the generic and RLD. But when the samples are analyzed by SEC, there's some sort of like a elbow-shaped curve, and indicating there might be some artifacts in the SEC analysis. So in that case, that we may wonder whether the similarity or difference between the generic product with the RLD is due to the nature of the sample or due to the artifacts during analysis. So in that case, that when the SEC showing some limitation, AF4 can be used as a primary method. So if SEC is proven to be sufficient in analyze this type of sample, we still can use AF4 as a orthogonal method to provide supporting information to SEC. So For protein and peptide molecule, a lot of time that they may be modified and to improve some of their property or to introduce new properties, and they may be conjugated with a different type of molecules. For example, protein can be modified with a PEG, and because of the signal response of a PEG is different from the signal response of protein and peptide in the AF4 detector, we can utilize this difference to determine the conjugation efficiency. How many PEG molecules on the protein, and by measuring the protein, the PEG, and the conjugate complex, and then we know the ratio between the PEG and the protein. So using the same strategy that we can also determine uses on the antibody drug conjugate, we want to know how many drug molecules conjugate to each antibody. So by using AF4, we can obtain such information. So beside the biopolymer that, AF4 can also be used for synthetic polymer. So here at OTR, we have utilized AF4 to study the high molecular weight polymer in the abuse deterrent formulation. So as we know that the solid opioid product often manipulated to achieve like an unintended route or for, defeat the extended release property. So the AD technology is meant to make the manipulation more difficult or less attractive. But the post-marketing data has shown there's a safety concern associated with this technology. For example, the Opana was once reformulated with a crush-resistant inner ingredient, but data show that there's a shift in the abuse route from nasal to intravenous injection after the reformulation. And this shift in the abuse route was accompanied by a sudden increase in the thrombotic microangiopathy. So it's a potentially a fatal blood disorder. And the study have showed that the high molecular weight PEO polymer is the possible cause of this TMA. So in addition, when we conducted an in vivo animal study, we also found that there's actually a molecular weight dependent toxicity for this polymer. For PEO that has a lower molecular weight, their toxic effect is much lower compared to the big PEO polymer. And also we know that PEO is not very stable and they may degrade under stress conditions such as heat and shear. And this type of stress condition is very common during the manufacturing process or during the manipulation. So, to better understand their toxic effect, we need to do a very good characterizations on this polymer. And among the different physical chemical property, the molar mass distribution is one of the critical one that affect the role of PEO in the ADF formulation. So we use the AF4 and SEC to study the molar mass distribution. As you can see that, the SEC has shown some limitation and difficulty for such big polymer. We see some co-elution of a big polymer with the smaller polymer. But for AF4 that, because it's much better suited for the big polymer or particles. And we use AF4 to analyze several different in-house placebo ADF formulation. They manufacturing under different condition and they further manipulate under different condition. And AF4, can easily capture the small change in the molar mass caused by the difference in the manufacturing and manipulation process. So this showing the advantage of AF4 over SEC in analyzing such big polymer. So actually more important is, when we develop this method, we found it's not really just restrict to PEO. It can be readily applied to other type of big polymer, that are commonly used in the drug product like HPMC or PVP. So the last example I want to show you is the analysis of solid dosage form. I mean, in most of the conditions, of course, we cannot directly analyze a solid tablet by using AF4 in situ. but we can use AF4 to study the component of a solid dosage form. So here we have some formulation made with a PLGA particle of different size. And so the AF4 is used to analyze the dissolution sample. And the benefit is that we don't have to do any sample treatment. We can directly inject the sample into AF4, and we can determine the amount of PLGA particle released from the tablet. And there are apparently some difference for the smaller compared to the bigger one. So far, I talked a lot about the application, and next I want to discuss a little bit of the deficiency when using this type of technique. So I mean, so far this AF4 is, even though they are very powerful and versatile, it's still quite quite new, and they're kind of a steep learning curve for this type of technique. And sometimes we notice that even the sponsor, they did not have a very good understanding of this technique. And when they try to use this technique to characterize their product, that will cause a lot of different type of deficiency they didn't even realize. So the first deficiency we have noticed in the application is the insufficient separation. So in AF4, there's always a peak called void peak at the beginning of the elution. So this void peak contains small amount of unseparated sample, the one that directly elute without any separation. So, I mean, this void peak is just related to the intrinsic nature of this technique, and there's no way to completely eliminate it. So what we can do is to have your sample peak separated from this void peak as much as possible. So you see the top figure is in their original submission, the sponsor want to utilize this method to study the high molecular aggregate in their peptide drug. And our comment is that your sample peak is not very good, separate from the void peak. That in that case is not a reliable method for the quantification of a high molecular weight aggregate. So we asked them to conduct a further development and optimization. In the amended submission, that the sponsor came back with their new method. As you can see here, the improved method is much better. The sample peak is almost baseline separated from the void peak. In this case, it's actually suitable for quantification purpose. So the second efficiency we have seen in the application is the run-to-run variation. As you can see here, the sponsor compared the molar mass distribution of their product compare them with the RLD. I mean, for the end product, we see some very run-to-run variation and some slight difference in the molar mass. So actually, this type of variation is not uncommon and is in general acceptable. But when you look at the peak retention time of the ANDA and compare them with RLD, you can see a big difference. The peak retention time shift from 10 minutes to around 8.5 minutes. And so such big change in the peak retention time indicates either whether you have changed your analysis condition, or there's some change to the sample that affect their the result. So we asked the sponsor to provide explanation for this result. So they came back claiming that they did not analyze the end sample at the same time as an RLD sample. Actually, it's a few months apart. So they think it might be due to the membrane aging. So in their new submitted data, they analyzed both of their proposed generic product with the RLD in the same day, and the new data show that there's a very small run-to-run variation, which is actually acceptable this time. So the third deficiency we have seen in the application is the inconsistent data processing, which actually important here. here, I mean, you can tell that, the peak width of this green color one is quite different from the rest of the two analysis. It's I think these are 3 repeat run for the same samples. So in that case, that when you change your peak width, I mean, we have a question, whether the sample within this selected peak width is a meaningful representation of your sample. Does it reflect the majority of your sample population? So we asked the sponsor to provide the explanation on this discrepancy. And so they came by with an explanation claiming that, they use a manual selection mode, so in their new submit data, they let the software to automatically determine the peak weights and by giving its criteria. And in their new submit data, the peak weights and is more consistent. So, I mean, next, I will use the last few minutes to talk a little bit about several other techniques. Today our focus is mainly on the asymmetric flow, field flow fractionation, and we know that there's several other FFF technique in the FFF family. So in general, no matter what they are used, what separation mechanism they are based on, they always rely a liquid stream to flush the sample out to the channel into the detector. So that's why they are all called flow fractionation. And so the difference are, what mechanism are they used to generate this separation field. So the first one is called the electrical AF4. As you may notice that the electrical FFF is, in terms of the channel design, quite similar to the regular AF4. They adding two electrode to the channel. So in this electro AF4, in addition to the liquid flow generate the separation field, we have a electric field. So the particle separation here is not only based on the hydrodynamic size, can also be based on their surface charge difference. And here I'm showing analysis of a liposome formulation by using electrical AF4. So when we do not apply a voltage, and the analysis will be just the same as a regular AF4. And so in regular AF4, that this liposome formulation seems to be, fairly uniform. But when we start to apply voltage, there's a shoulder peak start to emerge. So that means that despite there's some uniformity in the hydrodynamic size, there might be some difference in their surface composition that result in a different amount of surface charge carried by the particle, that result in shoulder peak. So electrical AF4 is very sensitive to pick up such a subtle difference in the formulation. So next one is called the sedimentation FFF. So for this one we don't have a flash-shaped channel. Instead, there's a ribbon like a channel is folded into a circular shape. They will rotate around a rotor, and the field is generated by the centrifugal force. So in this case, heavier or denser particle under this field will stay to the outer layer of the channel, and the smaller particle, they will stay relatively closer to the center of the channel. And the channel flow still has a parabolic profile, and in that case, a smaller particle, they will experience faster channel flow, so they will be eluted at the early time. So this technique is very useful to analyze the relatively bigger particle, particle a few hundred nanometer in size or even micron size, or some very dense particle like metallic or metal oxide particles. So the last one is called the thermal FFF. So this technique, the field is generated by the temperature gradient. There's two, the top and the bottom channel block has different temperature. And so the liquid flow is only respond to flush the sample out of the channel. And so this will be used to analyze samples that may have they have different thermal diffusion, and thermal diffusion will depends on the chemical composition of the polymer or the branching status of the polymer, whether they are linear or branch. Okay, so far, I mean, I talk a lot about the characterization of using AF4, but you may notice that most of this property I talk about is relate to physical property, size, charge, density, and but for a complex drug that we want to know more. Not only we want to know the size distribution, we also want to know, how much API is loaded in each size fraction, and whether there's any variation on the excipient profile in the smaller compared with the bigger one. And for the chemical composition information, in most of the cases that people use LC, UV, or LCMS, but if they are used alone, that in general, we can only know the total amount of a certain compound. And in order to correlate the composition with the size, we need to couple two different techniques together, either online or offline. So, for example, that if we use AF4 as the first dimension to separate the liposome formulation, so the liposome will be separated based on their hydrodynamic size, and they will pass through a series of online detector. In that case, we know, okay, the particle size, the molar mass, their shape, and the concentration of each size fraction. And so each separated fraction will then enter the second dimension. So the second dimension, the separation mechanism should be different from the first dimension. So the first dimension is separate based on the size, and the second dimension should be rely on another type of mechanism. For example, rely the chemical composition different. So in that case, if we use LCUV as a second dimension, each tiny separated fraction will enter the second dimension and then will be analyzed. In that case, we will know the amount of drug in this size fraction and the amount of each excipient in this fraction. And then by combining these two together, we can correlate, okay, how much drug or how much excipient in the 50 nanometer with 80 nanometer. Here I'm showing an example and analyzing a liposome formulation. And by coupling AF4 with LC, that we can tell, okay, how much drug in the 80 nanometer liposome and how much drug is loaded into a 100 nanometer liposome. We can also tell whether there's any difference in the lipid profile, the amount of cholesterol in the big versus small liposome particles. So by coupling two separation techniques together, we'll get much richer information regarding the formulation. So last, I want to use this slide to conclude my presentation. We are building this advanced and separation and characterization lab, just dedicated to answer the question related to complex drug product. We can analyze a variety of different complex product, lipid particle, liposome, PLGV, antibody, therapeutic protein, nucleotide, and polymeric excipients. And we can measure very different physical chemical property. And by studying them, we can get a very comprehensive information regarding the formulation, the aggregation status and whether the formulation is stable, what are the particle size, and what are the distribution of API. So, and the bottom line is currently the information we have at OTR, the BQR. And so this will conclude my presentation, and I'm ready for any question you may have. Thank you.